So, tēnā koutou katoa, ko Gwen Grelet tuku ingoa, ko ka maitai, ko yora taku mahi I te manaki whenua Linkia Research. I'll be introducing this talk, which is really a joint presentation between myself and Andrew Dopheid, who's also online here. So we're going to talk to you about our contribution to the program, which our contribution was focused on soil biology, and particularly we tried to understand the role it played in the nitrogen and carbon cycling within the context of the experiments that Scott and William described to you earlier. And we wanted to understand that because we want to understand whether we can manipulate soil biology so that it responds better to intervention and management practices that we might decide to make. So we can reduce nitrate leaching or increase soil carbon, for example. So this presentation will provide to you some of the insight we obtained, not all of it, just to highlight. Most of this work was conceptualized between David Whitehead, strong input from Steve Wakeland and myself. Andrew, who will be presenting, is really led all of the data analysis and write-up. And then I'll also be speaking to some of the data obtained by Jonathan Nunes, who just completed his PhD supervised by David and Matthew, and just started a new postdoc in Switzerland and actually moved with his family last week. So really exciting new start for him. And I'll now hand it over to Andrew.
Okay, thank you, Gwen. Okay, so first of all, I'm going to talk about soil biodiversity data from the lysimeter experiment that was talked about earlier, specifically the ones where sucrose was added to soil with ryegrass and white clover vegetation at 3 different rates. And then the effects of those carbon inputs on nitrogen losses observed over time. So just these 3 treatments highlighted here. This showed that adding carbon did indeed reduce nitrogen losses. For example, this graph shows that urine with no added carbon, represented by the orange data points, we see or results in a large spike in soil nitrate concentrations over about 14 days, which then declines from about 28 to 56 days. In contrast, urine with added carbon, represented by the purple points, results in considerably lower concentrations of nitrate over the same periods of time. Now we know that these reductions in nitrate are attributed at least in part to carbon-stimulated microbial uptake of nitrogen. However, apart from that, we don't really know what happens to soil biodiversity when urine and carbon is added. In particular, we investigated these two questions. Firstly, do microbial communities in soil return to their original richness and composition over time, or are they shifted into a different state? Secondly, prokaryotes and fungi, which are both major components of soil communities, have contrasting biological characteristics such as differing growth rates. So do they show different responses over time to urine and carbon inputs? This gives an overview of changes in community composition of prokaryotes in the top row and fungi on the bottom row, and how they're affected over time by urine plus no carbon input on the left, or 2 different rates of carbon input in the middle and the right 2 boxes. The colors of the points in the figure indicate the time of sampling. So purple points are immediately before the urine was applied, and the yellow points are after 112 days has passed. And what we see is that the urine plus the carbon inputs result in much larger shifts in the community composition than inputs of urine alone, and with the biggest shifts occur between about 7 and 14 days. And we also see considerably larger shifts over time for fungi than we do for the prokaryotes, with communities appearing to be very different even after 112 days. This shows some more details about how the communities are changing. This shows the proportions of prokaryotes or fungi that were present at the start of the experiment and then persisted throughout the experiment, which is shown by the grey bars, or were lost throughout the experiment, shown by the yellow, or gained, which is shown by the blue, relative to the start of the experiment. And we can see that there are contrasting effects for prokaryotes and fungi. With no carbon input, there are moderate declines over time with urine only, but much more turnover, i.e. replacement of lost organisms with new organisms for fungi compared to the prokaryotes. Inputs of carbon, however, appear to minimize any losses over time for prokaryotes. Any losses are balanced out by gains. In contrast, carbon inputs appear to greatly exacerbate losses of fungal biodiversity over time, with no real sign of recovery after 112 days. So from this, we can see that prokaryotes appear to be more resilient to carbon inputs than fungal communities. Moving on to the next piece of research, which was focused on how biodiversity, nutrient cycling, and functions changes from the soil surface to the deep soil. Knowledge of soil biodiversity and nutrient cycling is quite biased towards the prokaryotes and the top 10 centimeters of soil. Of course, this is where plants, nutrients, and other resources are most abundant, and these resources become less abundant with increasing depth in the soil. So how do these changes in resource availability throughout the soil affect communities, especially eukaryotes, and functions at deeper levels in the soil? To investigate these questions, a collection of 18 deep soil cores, each one 20 centimeters in diameter and up to 1.7 meters deep, were collected in the stony soil from 3 different paddocks at Ashley Dean Farm. In the middle there is a picture of the soil coring process, which involved a hydraulic drilling machine, which apparently would more typically be used to assess subsurface structure for construction projects. And on the right is one of the extracted soil cores in the lab. Now from each of these soil cores, 3 different fractions of the soil were extracted. So what we call D1 was the top 10 centimeters. D4 was 30 to 50 centimeters deep. This was intended to be sort of the border of where plant roots would stop. And then D9 was as deep as we could possibly get, so 1.1 meters down to the maximum depth. The maximum depth actually varied somewhat between the cores because soil couldn't be recovered from the full depth in all cases. Then on each of these samples, a variety of soil chemistry and nitrogen cycle analyses were carried out, and we also did biodiversity analyses via DNA metabarcoding for prokaryotes, fungi, protists, and nematodes. At this point, I think Gwen is going to tell you about the nitrogen cycling at different depths, and after that, I'll talk about the biodiversity analyses.
Thank you, Andrew. Do you want to? Thanks. Right, so this work was one of the chapter of the thesis of Jonathan Nunes. So Jonathan wonderfully arrived, I remember, just one day before we started the deep coring and the processing of the cores, and he launched himself into helping us and designing a side set of measurements so he could ride the wave and the opportunity that was given by the collection and enormous effort that was invested in collecting these soil cores and processing them for DNA analysis. And so what Jonathan looked at was how is the concentration of nitrate in the topsoil and the subsoil linked with properties of the microbial community. And the reason for this was because you've heard earlier a lot about the fact that ashadin is set on stony soils and resources and water flow through to depths. We wanted to specifically understand, what really can happen at depths, and can we find management practices that are more actively intervening at depths, so we can catch any nitrate that might leach down. So what we did is, so Jonathan here showing you in the middle of that slide the fact that in the topsoil the mineralizable nitrogen pool is about 10 orders of magnitude bigger than what you find in the subsoil. So we also have a clear contrast in nitrogen abundance and the concentration of free-flowing nitrogen. Can you just move to the next slide? Yeah. So what Jonathan did is he measured microbial biomass carbon, which is a proxy for how much carbon is locked up in the microbes, and can also be interpreted as a proxy for the abundance of the microbial community. He's also measured the abundance using qPCR of key genes that are involved in the nitrogen cycle, so genes that are involved in a denitrification process, in the nitrification process. And he also measured the enzymatic activity of these genes, or that lead to denitrification. Is then applied a structural equation modeling analysis to tease out which of the variables that he measured were correlated with each other. And that's a way of understanding what drives what in the system. So here are the results of the structural equation modeling for the topsoil. So the first 10 centimeters of the soil, which is from 0 to 10 centimeters. And what he found is that the denitrification enzymatic activity is strongly correlated with the abundance of nitrifier genes, which in turn are correlated with microbial biomass carbon and ultimately with the abundance of mineralizable nitrogen. However, this biologically driven part of the nitrogen cycle was not at all correlated with the abundance of nitrate ions. So one interpretation of this data is that in the topsoil, nitrate concentration, and so remember these are well-drained topsoil, is independent of microbial properties. If you move to the next slide. So now looking at what happens in the subsoil. So as opposed to this, in the subsoil, pretty much everything is correlated. So you have the organic, the mineralizable nitrogen fraction, which is correlated with the abundance of both your nitrifiers and denitrifiers gene. The bacterial diversity is strongly influencing the mineralizable fraction. And then in turn, that influences the overall denitrification activity, which is then negatively correlated with the abundance of your nitrate ion. So one interpretation of this data is that in the subsoil, the entire nitrogen cycle is coupled with biology and the diversity of the microbe. The subsoil nitrate concentration is strongly driven by microbial processes. And basically management that alters microbial diversity in the subsoil could potentially increase denitrification and reduce nitrate leaching. Do you want to move to the next slide? Yeah, so I think now I hand it over to you, Andrew.
Yep, okay. So as we mentioned earlier, we looked at the biodiversity of prokaryotes, fungi, protists, and nematodes at these 3 different depths. I'll just note that we included protists and nematodes because these are both heterotrophs that eat other things, and they're thought to be important or have important roles in soil food webs, and we don't really know much about them in the deep soil in particular. 3 biodiversity questions that we investigated were, firstly, do all these different taxa show the same trends with increasing depth? Are communities in the deep soil a subset of those at the soil surface, or are they something completely different? And thirdly, how does the organization of communities, as indicated by network structure, change with depth and declining resource availability? We observed that overall, the richness of all groups declined substantially with increasing depth. When we looked at taxonomic subsets of each group, such as fungal classes, protist-phyla, and nematode orders, again, almost all of those declined with depth and none of them increased with depth. In contrast, only 13 of 25 prokaryote phyla declined with depth, and then we had 9 that increased substantially with increasing depth, which included 3 groups of archaea, as well as this bacteria and firmicutes. This shows the depth distributions of these different groups throughout the soil column, which differed between prokaryotes and eukaryote groups. We observed that about 75% of prokaryotes occurred at multiple depths, at 2 or 3 depths in the soil, including 40% that were detected at all 3 depths in the soil. In contrast, fungi, protists, and nematodes were more specialized by depth, with about 40 to 60% found only in 1 depth, mainly in the topsoil, and 10% or less found at all depths. So what this tells us is that there is still substantial biodiversity of both prokaryotes and eukaryotes beyond the topsoil. Only looking at the topsoil will tend to overlook much of the biodiversity that's present. And also farm management strategies, which may be largely based on what's in the topsoil and on bacteria, may not cater for deep soil biodiversity, particularly eukaryotes, which might in turn affect things like nutrient movement and loss throughout the soil. Next, we looked at changes in the trophic composition of nematodes at different depths, and we observed that plant-feeding nematodes are most common at the soil surface, where plants are abundant, whereas in the deep soil, bacteria-feeding nematodes are the most common, presumably due to a lack of plant material and an increased abundance of or availability of bacteria, relatively speaking, in the deep soil. So this illustrates that changes in the biodiversity throughout the soil do translate to changes in functions at different depths. And we can expect that different sets of organisms at different depths will affect how the soil ecosystem functions at these different depths. Another way to look at biodiversity is through co-occurrence networks, which tell us something about how communities are organized or structured. This shows networks from the topsoil and the deep soil. The topsoil network is clearly bigger and contains a wider variety of taxonomic groups than the deep soil network, which is dominated by bacteria. However, looking at this doesn't really tell us that much about how the network is organized. So instead, we used some statistical methods to analyze the structure of these networks. And this graph shows 3 different metrics of network structure. So we've got edge density, closeness, centrality, and modularity. And these tend to increase from the topsoil down into the deep soil, except for modularity, which increases from topsoil to the midsoil, then declines again to the deep soil. What this suggests is that communities in the deep soil tend to be more closely associated and interconnected than those at the surface. And this is likely because in the deep soil, nutrients and resources are scarce and probably more heterogeneously distributed compared to the soil surface. So communities in the deep soil are likely to be more concentrated around these scarce niches or deposits of resource availability, where they might potentially cooperate to meet their metabolic requirements to a greater extent than in the topsoil. In contrast, in the topsoil, nutrients are abundant and everywhere, and therefore communities in the topsoil are less structured and less likely to be organised into clusters of organisms. I'll point out that although modularity decreased, or was lowest apparently in the deep soil, modularity measures the tendency of a network to be split into small clusters of organisms. And although my modularity decreased in the deep soil, the modules or the clusters of organisms that the network broke into were bigger than the topsoil and the midsoil. So I think that graph doesn't quite give the full picture on that point. So this all shows that communities in the deep soil have different structure, composition, and likely function compared to those in the topsoil. And with that, I'll hand back to Gwen to sum up.
Thank you, Andrew. So what does it all mean? So the first thing we looked at is, you know, what can soil biodiversity, what role can it play in optimizing and leaching mitigation strategies, and with a particular focus on understanding their role in what was observed in terms of the impact of adding readily available plant carbon. And so, William and Scott, well, we need more science to continue to understand the potential. We can say that readily available carbon in the form of sugars reduce enriching following urine. But what we've observed is that this extra addition of carbon has a long-term impact on soil fungi, and so specifically. And so although within the kind of the 112 days when that experiment was conducted, we can observe a desired sought-after outcome, which is a reduction in leaching, we don't know what might be the long-term consequences. We don't know whether that change in biological community will then impact the ability of the soil microbes to process addition of nitrogen from following subsequent urine. And we don't know whether there's going to be a loss in important properties that could, for example, help with resilience to extreme weather events, or you know, changes in fertilizer application or anything, any changes subsequently. Have we lost the ability to be resilient to that and respond in the way that we want? If you want to move to the next slide. Yes. So the other thing that our work has exposed is that soils are really a living system, and they extend well below the top layers, which are usually the topic of the focus of the investigation, because that's where most of the roots grow, and it's also much more easy to study. As you saw, we went to quite a big effort, and there was a lot of resource invested in collecting these soils at depth. However, if we are really trying to understand and harness the full breadth of the capacity that the biological community can offer and work with it, there's a huge amount of biodiversity that we're completely ignoring by not looking at what's going on at depth. We showed you data that the topsoil, the subsoil, and the deep soil communities are different and they function differently. We've also showed you that biology and nitrate related processes are uncoupled in the topsoil, and that could be interpreted in a way that basically the way the system that we studied were managed suggests that an input could be further optimized and potentially reduced to further reduce nitrate leaching. Of course, that's a trade-off with production, the way these systems are managed. What this data are also showing is that grazing management and land use management that are designed to increase plant carbon input and return to the soil might have a potential for reduced leaching. And amongst these are loads of opportunities that we don't really fully understand from a scientific standpoint. So increasing the diversity of plants, and particularly adding different type of root system that root at different depths that include a different allocation of carbon from the above ground to the below ground. I remember some early studies on high sugar grass and how that might impact leaching. And so harnessing that kind of diversity and putting it in the species mix might lead to reduced leaching. And management such as the trampling that is being talked about in the media quite a lot, and with regards to regenerative agriculture, for example, and leaving higher residuals where you have a greater amount of biomass above ground and therefore greater likelihood of carbon allocation below ground, that also might reduce leaching, but we don't know because we haven't got the data to say that. And then there could be some other strategies that are specifically designed to change the microbial biodiversity and the diversity of the soil organism at depths that could be used that are much more based on direct intervention and could still be explored. And with that, we will leave it there and happy to take questions.